Do you know what CRISPR is?
No, what is it? I thought it had some new way of gene editing and it has something to do with cloning. I don't really remember. I know it's a big deal.
CRISPR was discovered as a component of a bacterial immune system. Today, scientists use it as a genome editing tool. CRISPR is exciting because it's faster, cheaper, and more accurate than previous methods of genome editing. It has expanded what scientists have been able to do as far as new experiments and is speeding up the rate of new discoveries. So what does this mean? By making research easier for scientists, this could lead to new discoveries that help cure cancer and other diseases. CRISPR lets us change our DNA in a way that allows us to do this. We just have to figure out what to edit first.
Look at this graph!
CRISPR was discovered in 2002, but its purpose as a bacterial immune system wasn't uncovered until 2007. Once it was adapted as a gene editing tool in 2012 and used in animals in 2013, its use has skyrocketed.
What?
So how does CRISPR work? When a virus infects a bacteria, the viral sequence is recognized and incorporated into the bacterial genome between repeated sequences, giving them the name spacer sequences. When these genes are transcribed into RNA, they combine with the Cas9 protein to create CRISPR-Cas9. Then, if a virus tries to infect the bacteria again, the CRISPR-Cas9 destroys its DNA. So let's take a closer look. First, the guide RNA contains a complementary sequence to the target DNA and base pairs to it. The Cas9 enzyme is an endonuclease, which means it cuts the DNA. If scientists can create a guide RNA, then they can cut the DNA wherever they want.
Why?
Using CRISPR-Cas9, we can make exact cuts in the DNA, making it easier to perform a large number of experiments, such as disrupting a gene or introducing a new gene. Additionally, by mutating the CRISPR protein, we can use it to locate a gene without cutting the DNA, or we can turn genes on and off to treat diseases. For example, we can put activating proteins onto Cas9 and have them target insulin genes to treat diabetes in mice. That sounds easy. We should use CRISPR in humans right now.
That is not correct.
Well, it's a little bit more complicated than that. While CRISPR is an extremely useful tool, the human genome is about 3 billion base pairs long, which means there's a large potential for CRISPR recognizing the same sequence somewhere else and making off-target cuts. Scientists must be absolutely sure that CRISPR won't accidentally hurt people before we can use it in medicine. However, there has been some cool breakthroughs using CRISPR. For example, the first human embryo was edited in the US to correct a heart condition. In China, they created Hercules dogs and also injected CRISPR-edited cells into a patient with lung cancer.
Wow.
As it goes with any scientific breakthrough, there are ethical questions to be considered. As of now, the United States still has not used CRISPR in humans. If we were to do it, should it only be used to treat diseases or also for the enhancement of desirable qualities, such as intelligence or height? We need to be careful since we don't know all the risks yet, but they are currently being researched.
What are they saying?
Today, scientists are researching CRISPR, its effects, its many potential uses, and what genes to edit with CRISPR in flies, mice, and other model organisms so that someday soon we can hopefully use this in humans and be able to cure cancer or other diseases.